this video, I want to do a summary of all the processes of respiration. And this includes glycolysis, the link reaction, Krebs cycle, and oxidative phosphorylation. If we get a chance, we might even try and fit in a little bit of anaerobic respiration too. It may not be as detailed as the separate processes that we've looked at, but hopefully it'll give you a better idea of how these different processes actually work together and where they take place in the cell. We're going to start off with glycolysis. And we start off with glycolysis with glucose. Glucose is phosphorylated using ATP to form ADP and glucose phosphate. That glucose phosphate then is turned into triose phosphate, and because glucose phosphate is a 6-carbon compound, glucose is a 6-carbon compound, triose phosphate is a 3-carbon compound, so we get 2 lots of triose phosphates formed. Then triose phosphate undergoes a series of chemical reactions, and some of these involve other molecules. So we get inorganic phosphate being added, and then we have an oxidation taking place. So we have 2 NAD coming in. They remove hydrogen from the triose phosphate to form 2 reduced NAD. And these are very important, as we already know. The next stage is that 4 ADP come in to form 4 ATP, which of course is also very important. Now this is a form of substrate level phosphorylation, because here ADP is being phosphorylated at this point where these reactions are taking place, and not in oxidative phosphorylation, which we'll see a little bit later. So once we've had all those reactions taking place, we end up with 2 molecules of a substance called pyruvate, and that is also a 3-carbon compound. That is glycolysis. So please remember that this is a phosphorylation here of glucose. We sometimes call this a lysis or a lytic reaction, because the 6-carbon compound is broken down into 2 3-carbon compounds. We have a bit more phosphorylation here, and then we have got an oxidation or a dehydrogenation, the removal of hydrogen. Then we have a bit of ATP formed from ADP and the phosphate groups that had been added here. And we end up with pyruvate. So this process we would call glycolysis, and this takes place in the cytoplasm of the cell. Now pyruvate can go one of 2 ways. If there's no oxygen present, it can go to anaerobic respiration, which if we have time I'll put down here. But if oxygen is present, pyruvate is actively transported into the matrix of the mitochondria. So pyruvate is actively taken up into the matrix of the mitochondrion. And it initially undergoes a reaction called the link reaction. And at this stage, pyruvate is converted into acetyl groups. In doing so, two processes take place. First of all, NAD comes in to form reduced NAD. So there we have an oxidation. And we also have the loss of carbon dioxide. And that is a decarboxylation. So this process is called oxidative decarboxylation. And that is link reaction. Now the only other bit to the link reaction is that this acetyl group, which if you think about it, this had 3 carbons in, it's lost a carbon here, so that must be 2 carbon. This acetyl group cannot exist as just acetyl groups. It has to be attached to something. So what comes in at this reaction is a substance called coenzyme A. So it's a coenzyme, and its job is to pick up acetyl groups. So these acetyl groups are attached to something and therefore can be used. So we end up with a substance called acetyl coenzyme A. And that is the link reaction. The next stage is that acetyl coenzyme A then enters Krebs cycle. So the acetyl coenzyme A splits into acetyl groups and the coenzyme A. And that coenzyme A can then go back to another link reaction and take an acetyl group from another pyruvate molecule. That acetyl group here now joins on to a 4-carbon compound which is present in the matrix. So all this is happening in the matrix, and you'll see when I draw the next bit that is where it's happening. So this acetyl group is 2 carbon. It joins to a 4-carbon compound, which you don't need to know the name of. And that, of course, will form a 6-carbon compound. That is then converted into a 5-carbon compound. And to do that, first of all, it loses a bit of hydrogen to form reduced NAD, which is important, and it loses some carbon dioxide as well. And of course, the result of that is that this molecule is a 5-carbon compound. This is the same as that process, oxidative decarboxylation. And to make things nice and easy, the same thing happens again. This time we have NAD forming reduced NAD. Getting a little bit short space here, but I'm sure we'll be fine. And we also get carbon dioxide release here. So this is another oxidative decarboxylation happening twice there. Then we have a number of other reactions. This 4-carbon molecule can form into another 4-carbon molecule. And when it does that, it's able to phosphorylate ADP and inorganic phosphate, so they can join together to form ATP. This, like here, is another example of substrate level phosphorylation. All that means is ATP is being formed. So ADP is being phosphorylated here at the substrate of the enzymes that are involved in these reactions, and not at oxidative phosphorylation. Okay. Then that 4-carbon compound will be converted into another 4-carbon compound. And for this to take place, it loses hydrogen. And this is where FAD comes in to form reduced FAD. Why it's FAD here? It's just that NAD can't be used, so FAD is here. At least some of the hydrogens are being removed, at least some of the energy is being transferred. And then finally, that 4-carbon compound forms that one, and in doing so, it loses a little bit more hydrogen to form reduced NAD. So this time it is to NAD. And so we have Krebs cycle, or Krebs ovoid, because it's not quite a cycle. So you can see here how all these processes are linked together. They're all linked together. And if we go back to glucose, glucose contains a lot of chemical potential energy. And this process is basically to take away molecules that have that chemical potential energy. And in this case, those molecules will be hydrogen. So we've got hydrogen coming off here, hydrogen coming off here, 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 and here. Remember that from one glucose molecule, 2 pyruvate molecules are formed. So everything here happens twice. That's really important. So I'm not going to put 2 times on everything here, because it would just make everything very, very compact and confusing. The final part of aerobic respiration is when these reduced coenzymes are used. And these are used, and this takes place on the inner mitochondrial membrane, using carrier proteins in that membrane to pass hydrogens and then electrons between them. So I'm not going to draw the traditional diagram that I did last time, but rather I'm going to show you what is in the membrane. I'm also not going to be able to draw the membrane fully, because that would be far too complicated. So in the inner mitochondrial membrane, there are proteins which can take up or accept hydrogen. So there's one protein. And there's another protein. And let's get these reduced coenzymes moving across to here. So this is where we might start. It might be a little bit difficult to draw, But basically, all these reduced coenzymes move to the inner mitochondrial membrane. So I'm just going to have these joining on to here. Like so. And reduced FAD here is a little bit different. So we're going to have that coming off in a bit. So all these reduced NADs and the FAD move to the inner mitochondrial membrane. And let's start off with the reduced NAD. So the reduced NAD will give hydrogen to that first molecule. Then it will pass that hydrogen to the second molecule. After that, there are some more proteins in the membrane. I'll draw them slightly differently. And what happens at this point is that the hydrogens are separated. So these are hydrogen atoms. And so they are separated into hydrogen ions and electrons. So hydrogen ions come off here, and the electrons then pass to that one. Then they move from there to there, and those electrons then move from there to there. Now, importantly, what is happening here is a series, I'll just put NAD back up there. What is happening here is a series of oxidation and reduction reactions. So as the NAD loses hydrogen, it is being oxidized, and as this hydrogen protein carrier picks up the hydrogen, it is being reduced. When it loses it, it's being oxidized, and then that one is being reduced. So we have a series of reduction and oxidation reactions, like so, all the way along. The same thing happens with the electrons. So when these carriers pick up electrons, they are reduced. We say they are chemically reduced. When they lose the electron to the next one, we say that they are chemically oxidized. Okay? So that is the process up to this point. The only thing we need to think about here is the reduced FAD. So I'm going to make the reduced FAD come off here, because for various reasons the reduced FAD can't give its hydrogen to the first carrier. It can only give its hydrogen to the second carrier. So FAD is a little bit of an oddball, really, because it can be used here and it gives it to the second carrier like that. So that's great. So that there is the electron transport chain in the inner mitochondrial membrane, which I'll talk about in a moment in a bit more detail. Now, what we end up with here are hydrogen and electrons. That's no good, really. They need to be removed. So, of course, we now know that oxygen comes in, and the oxygen will pick up the electrons, will pick up these hydrogens to form water. So if I just put a 2 in front of there, I'll just make that oxygen atom. then we can see that we get water formed. So oxygen is the final electron acceptor, and it makes sure that all these processes are taking place. So like a production line, if this last carrier cannot lose its electrons, then this one can't pass the electrons to there, and that can't pass it to there, and this grinds to a halt. And in fact, all of this process grinds to a halt. Even this one would, but we'll talk about anaerobic respiration if we get a chance. All of these processes grind to a halt because this can't be lost. So in fact, that's why oxygen is so important, and it allows, the presence of oxygen allows the hydrogens to be removed all the way through the process, bit by bit, and therefore we can access more energy to make the ATP. Talking of which, what about ATP? Okay, so now I'm going to try and draw the inner mitochondrial membrane. So this is coming down here, like so. So all these proteins are in the inner mitochondrial membrane, which just so happens is being a little bit twisty here, like so. So that's the inner mitochondrial membrane. I'll just draw a couple of phospholipids so you know that is the case. So this is the inner mitochondrial membrane. And remember, it is impermeable to hydrogen ions. That's really important, which we'll talk about in a minute. I'll just put a few more phospholipids here just to make us still aware that this is still the inner mitochondrial membrane. So we've got the inner mitochondrial membrane here. This is the inner mitochondrial membrane going all the way along, like so. All the way along here. And what's happening is, as the hydrogen moves from there to there, a little bit of energy is released. And then as the hydrogen moves from there to there, or rather the electron moves from here to there, after it's split from the proton, the hydrogen ion, a little bit of energy is lost. Not that useful here. But then as the electrons move from there to there, a little bit of energy is lost. And as the electrons move from there to there, a little bit of energy is lost. So we're having energy being lost in a slowed, controlled manner. And that's the important thing here. Because what we have in the membrane up here are structures called hydrogen ion pumps. And what they do is this. This is the matrix, and in the matrix are hydrogen ions. They might have come from here, or there might be ones that are already present. Don't worry about what individual hydrogen ions and electrons are doing, because basically in the matrix there is a pool of hydrogen ions. They might have come from the reactions here, or they might have come from other chemical reactions that are taking place. So a hydrogen ion is just a hydrogen ion no matter where it comes from. So in the matrix are lots of hydrogen ions, and this energy here is transferred to this hydrogen ion pump, and it uses that energy to pump hydrogen ions from the matrix. So this is the matrix, into the space between the two membranes of the mitochondria. So up here, which you may not be able to see, I'll just tilt it slightly. So up here we have got the outer mitochondrial membrane going around like so, and I'll have to draw it a little bit thicker because I've drawn the other one a little bit thicker. So this is the outer mitochondrial membrane, just a few little phospholipids there just to make sure we know what we're doing. So this is the outer mitochondrial membrane. Like so. So that's going around the outside, like we say. And then this membrane, which is the inner mitochondrial membrane, has in it some other structures. Now these other structures are going to respond and use the hydrogen ions that have been pumped into there. So this here is a hydrogen ion pump, and it's actively transporting hydrogen ions from the matrix into the intermembrane space. And that space is really important because it's keeping lots and lots of hydrogen ions separate from all the others. So what we end up with is a high hydrogen ion concentration here, and in the matrix we have a low hydrogen ion concentration. Now in this membrane there are also some other, get the pens out of the way because this is a big bit, there are some other proteins, special proteins. These proteins are intrinsic proteins, and there's two parts of them really. One part is a channel. That channel is going to allow hydrogen ions to pass through it. At the other end is an enzyme. That enzyme is called ATP synthase. So that is embedded in that membrane. So a few more phospholipids there just to make sure we know that's a membrane. Okay, so that's the inner mitochondrial membrane here. And what happens now is that there's a very high hydrogen ion concentration here and a low one here. This membrane is impermeable to hydrogen ions. So is that one actually, but we're more interested in this. So this is impermeable to hydrogen ions. So the hydrogen ions cannot, because there's lots of them here, cannot move across that membrane. So because of the difference in concentration, this is a form of potential energy. There's lots of hydrogen ions here. They're desperate to move away from each other because they're positively charged, and also they want to move down their concentration gradient. So they move out through this channel, and this is a form of facilitated diffusion. That does say facilitated. And as they move across, they lose energy. And that energy is used by this enzyme here to form ATP. So of course, this process here is chemiosmosis. It's phosphorylating, it's using energy from here to move hydrogen ions from there to there to create an electrochemical gradient. And then as the hydrogen ions move down their electrochemical gradient, they release energy, which is used to form ATP. So this is phosphorylation. Here we are oxidizing the electrons and the hydrogens. So the whole of this process is called oxidative phosphorylation. So we've got the matrix here, we've got the outer and the inner mitochondrial membranes. So if I try to, and if I just raise my camera a little bit, instead of trying to be very sleek with my descriptions and nice and tidy, I'm just going to do it so it's more useful to you. So all this is happening inside the mitochondria. So this would be the outer mitochondrial membrane, and if I just make it thinner like so. This is the outer mitochondrial membrane, so that's the whole of the mitochondria. And then this here is going to be the inner mitochondrial membrane, and I've zoomed in on this bit, of course. I'm trying to draw a few cristae here, which is very difficult, but hopefully you get the idea. I can do a little bit better now. Nearly went too far then. So hopefully you get the idea that the link reaction and Krebs cycle are taking place in the matrix. So here we've got the matrix. And then oxidative phosphorylation is taking place on the inner mitochondrial membrane. It's a little bit messy, but hey, that's where it is. And it's cool. Everything's good. So that is aerobic respiration. But of course, sometimes there's no oxygen present. So I might be able to fit down here a bit of anaerobic respiration. So if there's no oxygen present, then the oxygen cannot take up the electrons here. So this grinds to a halt. There's no energy release because the hydrogen electrons are not being passed between the electron carrier chain. And so there's no energy release, so this can't take place, so we can't get ATP. But more importantly than that, in some ways, because none of this is moving, then the reduced FAD and the reduced NADs can't offload their hydrogen, which means we don't get NAD and FAD made, which can then go back to all these processes. The whole lot grinds to a halt. So if there's no oxygen present, cells still need to produce ATP. Well, all this is out really because it's so interacting with oxidative phosphorylation. The only chance there is glycolysis, and we do get a little bit of ATP made, not very much. It says 4 there, but overall it's still only 2 per glucose molecule, and there's about 38 made. So, you know, it's a very, very small amount. So, what happens? Well, what we need to do is to make more NAD, because if aerobic respiration is taking place and suddenly stop, we're basically going to run out of NAD. And if we run out of NAD because it can't be recycled, then glycolysis reactions will stop there. They can't proceed. And the problem is, the production of ATP occurs after the removal of hydrogen and the production of reduced NAD. If it was the other way around, it wouldn't matter. But these molecules have to lose hydrogen before they can carry on to the reaction that will make ATP. So what you need to do is for reduced NAD to lose its hydrogen. So we can have anaerobic respiration taking place, and this takes place in the cytoplasm. So all this here is cytoplasm. That's the mitochondrion, this is the cytoplasm. Now, here is reduced NAD. And what happens is, there's 2 types of anaerobic respiration. So pyruvate can convert itself into ethanol, which then produces ethanol when reduced NAD loses hydrogen. So this is the process that takes place in plants and fungi. And we call that the ethanol pathway. So that's one type of anaerobic respiration. I'll just move the camera down a little bit there. It's getting a bit dodgy, but never mind. Okay. So that's what happens in plants and fungi. Pyruvate, it's converted into ethanol. That picks up hydrogen from the reduced NAD to form ethanol. But one aspect of that, which is quite important to remember, is that for pyruvate to be converted into ethanol, it needs to lose carbon dioxide. Okay, and then another way is pyruvate can be converted straight away into a substance called lactate. So we call that the lactate pathway, and that takes place in animals only. But for pyruvate to be converted into lactate, it needs the reduced NAD. So I think if I try and draw that round there, it's going to be quite complex. And you get the reduced NAD that will be made here, giving its hydrogen to pyruvate to form lactate. Now, the important thing here is that no carbon is being lost, and therefore this lactate can build up. But when oxygen becomes present again, it can be reversed. So this is a reversible reaction. Here, this is not reversible because we're getting rid of carbon dioxide, but here it is reversible. So that is the whole process of respiration. And so the different parts we have got here: glycolysis, here we've got the link reaction, which is in the matrix of the mitochondria. Here we've got Krebs cycle. And then here we've got oxidative phosphorylation. And these here are often called the electron transport chain. And this, of course, is chemiosmosis. Electron transport chain. And this is chemiosmosis. I hope that's been very useful. And if I try and zoom out a little bit more, there we go. Thank you very much.